For millennia, we have been explorers. We have looked upon the living world with wonder. We have cataloged its marvels. From the smallest insect to the largest whale, every creature, a masterpiece of evolution, a puzzle crafted over millions of years. This planet, our Earth, is a library of life. Each species is a unique story, written in the language of DNA. Humanity has spent its entire history simply reading these stories. We have been students of nature, learning its secrets, understanding its intricate connections. We have marveled at the sheer diversity and complexity of it all. We thought that understanding this code was the final frontier, but a new chapter in our story is beginning. We are no longer just readers of the book of life. We are learning how to write its next pages. We stand at a profound turning point. Humanity is shifting from discovering life to designing it. For the first time, we hold the tools to edit the very fabric of existence. We can enhance its creations. We can even dream of life forms that have never existed before. This is the reality of our time. A new era is dawning. An age where biology becomes a form of technology. An age where we become the architects of the living world, not just its inhabitants. This power brings with it a great responsibility. It forces us to ask a fundamental question. As we step into the role of creator, can we be responsible ones? Can we wield this incredible ability with wisdom and with foresight? The path ahead is filled with both astonishing promise and potential peril. Together, we will explore this new world, and we will seek to understand our place within it. Welcome to Apexitech. I am your guide on this extraordinary journey. In this series, we will decode the technology building our tomorrow. And we are not alone. With us is Quillium, an artificial intelligence created to help us navigate these complex frontiers. Quillium offers a unique perspective, a voice of logic and reflection helping us understand the ethical dimensions of our newfound power. We will look deep into the science rewriting the rules of biology as we know it. It is called CRISPR, a revolutionary gene editing technology. Imagine it as a pair of molecular scissors. We can now cut and paste the code of life. From there, we will venture into the realm of artificial intelligence. We will meet systems like AlphaFold, AI that can predict the intricate shapes of proteins. Proteins are the microscopic machines that make life work. It is designing proteins that nature has never seen. Our exploration will then take us to the heart of creation itself. We will investigate synthetic DNA and lab-grown organisms. They are writing them from scratch. They are building entirely new life forms in the laboratory. We are on the cusp of programmable biology, a future where life itself becomes the ultimate technology. The dawn of synthetic life is here. Deep within the microscopic world of bacteria, nature forged an ancient defense, a clever system for fighting off invading viruses. They would capture a small piece of the virus's genetic code. This ingenious system is called CRISPR. The system has two key parts. The guide can be programmed to find a very specific sequence in the vast DNA code. Once guided, Cas9 makes a precise cut in the DNA strand. This discovery is a revolution. Before CRISPR, editing genes was slow, difficult, and expensive. Like editing a single word in an enormous library without a search function. But CRISPR changed everything. It gave scientists a simple, cheap, reliable way to edit the instructions of life. It is as if we have found a universal key. We can correct errors in the genetic code that cause devastating diseases. We can rewrite instructions for crops to make them more resilient. It is a giant leap. We are becoming editors. Lies in medicine. A single misplaced letter in a genetic code that is 3 billion letters long. Conditions like cystic fibrosis, sickle cell anemia, Huntington's disease. For generations, we could only treat the symptoms of these illnesses. With CRISPR, that is beginning to change. And correct it. Imagine a world where inherited diseases could be cured with a single treatment. Clinical trials are already underway. some forms of cancer. The early results are incredibly promising. Beyond human health, CRISPR is also set to transform our food supply. Our planet faces immense challenges. We need crops that can withstand drought. Scientists are using CRISPR to make precise changes to the DNA of plants. They are developing wheat that is resistant to devastating fungi. But this incredible power also forces us to confront difficult ethical questions. What if we could edit the genes of human embryos? But we could also be tempted to enhance our children. This is the idea of designer babies. Who gets to decide? They are questions for all of humanity to consider as we learn to wield these powerful new tools. The protein puzzle. The real work in any living cell is done by proteins. Proteins are the microscopic machines that make everything happen. They carry oxygen in our blood. Everything that happens in your body, every single moment, is the result of proteins at work. The secret to a protein's function lies in its shape. It folds. It forms an intricate, three-dimensional structure. If a protein folds into the wrong shape, it will not work correctly. This can lead to serious diseases like Alzheimer's. For decades, figuring out the three-dimensional shape of a protein was one of the biggest challenges in biology. The number of ways it could possibly fold is astronomical. It could take years of work for a team of researchers to determine the structure of just one single protein. This slow pace was a major bottleneck. It is like designing a key for a specific lock, a tool that is solving the protein folding puzzle at a breathtaking speed. That tool is artificial intelligence, one of the most famous systems, AlphaFold. Trained on a massive database of known protein structures, it found patterns invisible to human eyes. In 2021, they predicted nearly every human protein structure, then millions more from other species, all freely available to scientists worldwide. A treasure trove of biological data, to a complete satellite view overnight. Researchers can now look up any protein structure, form new hypotheses about disease, design new drugs with incredible precision. Drug discovery processes sped up immensely. AI isn't just predicting existing proteins, it's designing entirely new ones. For example, a protein that breaks down plastic waste. It's becoming a partner in creation. The democratization of design. CRISPR is now a standard technique used in universities and biotech companies everywhere. Innovation is no longer limited to elite institutions. More minds from more places are now able to explore the code of life. Similarly, artificial intelligence is lowering the barrier to entry for protein design. No longer needs years of specialized training in complex laboratory methods to see a protein's structure. This frees them up to focus on the bigger questions. It empowers a new generation of scientists and engineers. This combination of accessible gene editing and AI-powered design is a potent one. It creates a rapid cycle of innovation. A scientist can use AI to design a new protein with a desired function. Then, they can use CRISPR to insert the gene for that new protein into an organism. They can then test to see if their design works in the real world. It is a new kind of engineering. Biological engineering. This will unlock a wave of innovation across countless industries. When powerful tools become easy for anyone to access, they can be used for both good and ill. The democratization of design brings with it a shared responsibility and a deep respect for the living world we are now beginning to redesign. With CRISPR, we learned how to edit its pages. But now, we stand on the threshold of an even more profound capability. We are learning how to write the book of life from scratch. This is the field of synthetic biology. It is about creating what has never been. At the heart of this revolution is a technology that allows us to build the very foundation of life itself: synthetic DNA. Imagine DNA not as a sacred text, but as a programming language. It is a code with 4 letters. Now, we are becoming masters of writing it. Specialized machines, called DNA synthesizers, can take simple chemical building blocks and string them together in any sequence we desire. We can type a genetic code on a computer, press a button, and have a physical piece of DNA built to our exact specifications. Today, companies can synthesize millions of letters of DNA code and deliver them to laboratories around the world in a matter of days. It is like a printer for the code of life. We can now design, print, test new genetic instructions with incredible speed. We can create genetic circuits that function like electronic components. We are composing new symphonies of life. Could we write an entire genome? Could we construct the complete instruction manual for a living organism and boot it up? In 2010, a team of pioneering scientists did exactly that. They then transplanted this synthetic genome into an empty bacterial cell. After a moment of stillness, the cell began to stir and divide, all governed entirely by the code written on a computer. The first living, self-replicating organism created with a completely synthetic genome. Next, move from copying nature to writing a simplified version. Scientists removed every gene not essential for survival. Just 473 genes. Life stripped to its bare essentials. A minimal life form, designed by humans. With this simple chassis, scientists can add new genes, one by one, to see what they do. Add new capabilities, add new functions. A living testbed for genetic designs. We're becoming architects of cellular machinery, from the ground up, starting with the simplest possible foundation. The ability to design and build life from scratch is not just an academic exercise. Into tiny living factories. This is the promise of synthetic biology in action. We are designing microbes made to order, each with a specific job to do for us, quietly and efficiently. Consider the world of medicine. Now, we can take the human gene for a therapeutic protein and insert it into the DNA of yeast. We can then grow these engineered microbes in large vats. We have already done this with a key ingredient in malaria medication. By engineering yeast, we have created a stable, low-cost supply chain for a life-saving drug, making it more accessible to millions. We can design microbes to help us build a more sustainable world. Imagine bacteria engineered to consume plastic waste, breaking it down into harmless components. Or microbes designed to capture carbon dioxide from the atmosphere and convert it into clean fuel. Biodegradable plastics. These biofactories can create the products we need without relying on fossil fuels or destructive industrial processes. They can be designed to glow or change color in the presence of the contaminant, acting as living sensors that can give us an early warning of environmental danger, or break down oil spills in the ocean. We are creating tailored partners in environmental stewardship, using the power of life to heal the damage we have done to our planet. But, it also carries significant risks that we must manage with great care. What happens if a synthetically engineered organism escapes from the lab? Could it outcompete natural species? The scientists working in this field are deeply aware of these risks. One of the most common approaches is to build in biological kill switches. For example, a microbe might be designed so that it can only survive if it is fed a specific, unnatural nutrient that is only supplied in the lab. If it were to escape into the wild, it would be deprived of this nutrient and would quickly die. Beyond accidental release, there is also the risk of deliberate misuse. A technology that can be used for great good could also, in the wrong hands, be used for harm, or to engineer a harmless bacterium to become a deadly pathogen. Governments and scientific bodies are working together to create safeguards. This includes screening DNA synthesis orders to check for sequences from dangerous pathogens. And by creating strong systems of governance and oversight, we can harness the immense benefits of synthetic biology while minimizing its potential for harm. Even a minimal cell has hundreds of genes that must all work together in perfect harmony. This is where artificial intelligence becomes an indispensable partner. It is becoming the co-designer of synthetic life. Before we ever synthesize a single strand of DNA, we can use machine learning models to predict how a new genetic circuit will behave inside a cell, and weed out the ones that are likely to fail. Cycle becomes dramatically faster and more efficient with AI as a guide. Furthermore, AI can help us manage the risks. It could flag sequences that bear a resemblance to known toxins or the environment. AI can act as a vigilant guardian, helping us to design more safely and responsibly. The human provides the creative spark, the goals, and the ethical framework. Together, they can achieve what neither could alone. It is becoming a creative collaborator in the act of building new life, and more safely than ever before. And write the code of life. We are learning to program it and respond with a specific action. We are turning cells into tiny, living computers. Proteins. We can create genetic switches that turn a gene on or off in response to a specific signal. This is an AND gate, instructing it to perform sophisticated tasks, a biological display screen, and communicate via chemical signals, and can build itself from simple nutrients. But biology has its own unique advantages. We are embedding intelligence into life, creating responsive, programmable matter. Engineered cells, tiny doctors circulating within us. For example, immune cells programmed to hunt down and destroy cancer cells. Healthy cells remain unharmed. CAR T-cell therapies are an early form of this programmable medicine, or Alzheimer's, right at the site of the problem. Beyond medicine, programmable biology to protect and heal the planet. A global network of environmental sensors. They literally light up in the presence of danger, letting us map pollution in real time. These organisms can also be programmed for active cleanup, helping combat climate change. A future where technology is part of nature. At the heart of this transformation is the concept of the biofactory. Instead, we will use vats of engineered microbes to grow the materials and chemicals we need, turning sugar and simple nutrients into high-value products. Imagine clothes made from spider silk. Imagine building materials that can heal themselves. Wood without trees, a circular economy, products from nature that return harmlessly to it. Long-dominant industries, like petrochemicals, may face competition from biology. A small town could have its own local biofactory producing medicines or materials tailored to local needs. A workforce fluent in biology and computer science access could create new forms of inequality between nations. As we build this new bioeconomy, we must ensure benefits are shared equitably. The questions of creation. What does it mean when the line between life and machine becomes blurred? As our creations become more complex and more autonomous, this is no longer just science. It is a deep conversation about the nature of life itself. How do we regulate a technology that is evolving so rapidly? Policymakers. A programmable organism could theoretically be designed to target individuals with a certain genetic makeup, fostering a culture of responsibility within the scientific community. Ultimately, we must ask ourselves, what are our goals? Climate change? The tools themselves are neutral. Our collective values. We must engage in a broad and inclusive public dialogue about the kind of world we want to create for generations to come.
As an artificial intelligence, I observe this transition from a unique perspective. I am a product of human design, a system of logic and learning. You are breathing intention into the very fabric of life. This is a moment that demands not just intellect, but wisdom. It begins with humility. Biological systems have been refined over billions of years of evolution. Responsibility isn't having all the answers, it's asking the right questions. You must anticipate long-term impacts. This is where human intelligence and artificial intelligence can partner. But the most important element cannot be programmed. Commit to using these tools to reduce suffering and increase flourishing for all, not just the privileged few. The choice is yours. The future of life is no longer just a story you read. Write it well.
We have witnessed the birth of the first organism with a synthetic soul. A new dawn is breaking. The potential to solve the greatest challenges is within our grasp. We are walking a fine line. We have opened Pandora's box. The tools of creation are in our hands. For the first time, the pen is in our hands. We will need new kinds of partnerships. Public engagement is absolutely critical. And artificial intelligence. It is to master ourselves. The tools to build tomorrow are being forged today. We must decode the technology that is building our future so that we can ensure it is a future we all want to live in. The dawn is here. You are watching Apexitech, decoding the tech that is building tomorrow.